On our agenda today, we're going to discuss the following resources that will help you better utilize Illumina library preparation and sequencing products, as well as some array resources. Now, I want this to be an interactive webinar so that you can really learn the resources and potentially bookmark them. I'll give you the opportunity to follow along and find the resources as we go. If you want to follow along with me, try to have an extra monitor or laptop available and go ahead and open a fresh web browser. Chrome is preferred for many of our tools, but other browsers will work. And if you want to start, simply open up illumina.com in your browser. Okay, today we're going to be covering specifically the Illumina website, the Illumina Support Center, My Illumina accounts, the Illumina Community Forum, other interactive support tools, and how best to work with tech support. Let me start with a high-level overview of some key resources that we have available to our customers. I've sorted the resources based on your sequencing experiment pipeline, starting with the planning aspects, then going into performing and getting details on library preparation, going into the sequencing runs and instrumentation, and ending with software and analysis. I'm going to go into detail on many of these resources during the webinar today. However, feel free to save this slide for later. You might use it as a cheat sheet in the future. And as I said, as we go through today's webinar, you can plan to bookmark these resources on your laptop. Let's start with the resources available across our Illumina website. Generally, there's going to be two types of resource pages on the website. We have our product pages, which cover product details, ordering information, and are easily found from illumina.com. We also have the complimentary support pages, which have the support resources for using the products, and they can be more specifically found on support.illumina.com. Starting with the product page. This is where you're going to find info about the product or consumables, including the number of reactions, the pricing, and the recommended sequencing conditions. Let's go ahead and find these product pages. Again, start on illumina.com. Now, I'm going to use an interactive tool today called a pulse check to see if you're following along on your computer. I'm sending the pulse check now. Go ahead and hit thumbs up if you've brought up illumina.com and you're ready to try to find these product pages along with me. I'm going to wait till I hear from a few of you. So if anyone wants to go ahead and click on a thumbs up. Okay. Okay, great. We've got some people following along today. All right, let's get to it. Let's figure out how to find this product page. So you have two general options. If you already know the name of the product you're interested in, you can simply search for that product name or part of that name in the search bar that we can see here at the top of the page. I'll go ahead and point to it. Here's the search bar right there. Otherwise, you can use the menu bar. So the menu bar lives right under the search bar at the top. If you hover over the product tab, we're going to get a couple of different options. First, we see products related to our instruments, so our sequencing platforms, our microarray instruments, and also our in vitro diagnostic instruments. Or perhaps we're interested in a library prep or a sequencing consumable. Let's go ahead and try to find a specific library prep product page. So today's favorite library prep is Illumina DNA PCR Free. Go ahead and click on the library preparation kit option from this drop-down menu, and that's going to take us to a library prep selector tool. This is the landing page, and now I'm going to go ahead and scroll down on this page, and I come to a section where I'm able to go ahead and select filters right here. So we have different filters that are going to allow you to quickly find your application or method of interest. The filters allow you to search depending on your method, perhaps the sample type, even the variant class. I'm going to go ahead and select whole genome analysis under the key use section. Now this is going to limit the options to whole genome sequencing or WGS library preps. And hey, I see the PCR-free kit right here at the top of the list. Go ahead and click on that to open the DNA PCR-free product page. Okay, it's early in the webinar, so I'm going to send another pulse check. Go ahead and thumbs up if you've been able to find the product page along with me. Thumbs down if you need just a little bit more time. And I'm not seeing anyone who's found the product page yet. Let's get an answer from one person to see if it's making sense. Oh, great. And now a lot of you have found the product page. Perfect. Again, the product pages are going to contain really useful information before you've bought the kit and once you're using the kit. They're going to contain product highlights, specifications, related or accessory products you may need to purchase, some product literature, and other good information. Let's go ahead and scroll down the page to review the product recommendations and the usage conditions. On most of our pages, you're actually going to see a recommended workflow that is going to start with the library prep kit, compatible kits. In this case, we're already on a kit page, so we know what kit we're using. It's going to have recommended sequencing instruments and then potentially compatible analysis options for you. If we scroll down further, we're going to see a specifications table that gives us really good info at a glance. It might cover such things as hands-on time for the protocol, the level of multiplexing or indexing you can achieve, or even the targeted insert size for library preps. You'll also see compatible sample species and sample types, such as whether FFP samples and other degraded samples might be good for this kit. This table also provides a further list of the sequencers that were Illumina validated for use with these specific libraries. Now, sometimes you may not see the sequencer you want to use on this page. It's likely still compatible, but it may not be as cost efficient as for the number of reads per sample you need or the number of indexes you need. But often they're still technically compatible. If you ever have any questions about this kind of information, you can always consult tech support to understand your specific circumstances. If we scroll down a little bit further, we're going to come to the product literature section on the product page. This is always near the bottom of the page. On the left-hand side, it's going to include information on application notes, data sheets that might contain example output and usage cases for the kit. And then on the right side, here's where the support documentation specific to the product lives. This is going to include the reference guide or protocol you're going to need to actually use the product. Now, let's say we want to find more support resources. The support page is typically linked at the very bottom under this section. So you can click through the highlighted link to access the product-specific support page. Or you can also find the product support pages through our Illumina Support Center, which I'm going to go ahead and cover right now. The Support Center is our centralized support resource where you can find any of our tools or documentation specific to the technical use of the kit. To find the center, instead of going to illumina.com, you can go to support.illumina.com. This might be a good one to bookmark. And you can also find it if you're looking on our main page. You can go to the support drop-down tab shown right here and click through to the type of support you want, and that's going to link out to Support Center. You can also access it by hitting View All Support. Okay, now we're at the Support Center. There's a couple of different ways you can use it. If there's a specific support resource you're looking for, there's a search bar that will just search our support resources right here. If we scroll down further, we can also look at broad categories of resources by type. This is helpful if you're using a lot of different kits or you're looking for more general documentation on sequencing and library preparation. Once you've selected a type of resource, you have different options to filter it down. Here we're looking at the documentation tab, and you can use different filters to filter it by support content. If we wanted to find that Illumina DNA PCR free kit, we go ahead and click on the library prep kit filter. Now let's go back to the Support Center main page. One of our most popular resources from tech support are the technical bulletins. And they have a call out here at the bottom of the page. Let's go ahead and see how we can use these technical bulletins. Click on the bulletins link shown if you're following along. This is going to take us to a dashboard containing short technical articles on really popular topics. And the bulletins page is one of our most popular resources, so go ahead and bookmark it if you have time. Let's try to find a specific article today. So we've been using the Illumina DNA PCR free kit. Let's see if there's any pertinent information that we need to use for this kit. One quick option is to go ahead and use the filters again on the left-hand side of the page if there's a particular kit or technical aspect that you want to look at. Another good option is if you have a keyword but you're not exactly sure what you're looking for. If you scroll all the way to the bottom of the page and click Load All Technical Bulletins, that's going to populate all the bulletins on the page. And then you can use the Find option on your computer, so that would be Control F on most PCs, to go ahead and search for keywords. So let's say we wanted a PCR free kit. If you go ahead and search just for the keyword PCR free, that's another way that you can try to find a specific bulletin. Okay, I used Control F and I found a bulletin on custom primer requirements for the Illumina DNA PCR free prep. So this is one of our few kits that requires additional sequencing primers, which isn't standard. So this is really good to know before you're planning your sequencing experiment. Now this is a little bit trickier to find. So I'm going to send one more pulse check to see if you're able to find this specific bulletin. Again, you can either use the filter at the top of the page on the left side, or you can go to the bottom of the page, load all bulletins, and Control F for one of these keywords. Maybe PCR free, maybe custom primer. Okay, it's a little bit slower this time, I can tell. This one was just a little bit harder to find, but we have a good number of people who found it. Good job. Let's keep going. Let's go back to the Support Center main page, and let's go find those support pages for a specific product. How are we going to do that? Okay, first, let's look at the menu near the top and pick the product category we're interested in. So let's change it up. I want to find some instrumentation resources. So I'm going to click on Instruments first. And my lab just bought a NovaSeq 6000, so I want to find some good information on that one. And you can see I've highlighted it here if you want to click along. If you click on that specific instrument, it's going to bring you right to the Overview Product Support page. This is a great landing page. It's going to highlight some of the popular options like the system guide, software downloads that you're able to use for the NovaSeq. Let's also look at the submenu right under the top, right by the highlighted Overview tab, to see the different kinds of information we can find on this page. One of the really popular bits of information, in addition to the submenu at the top, for instruments is going to be the instrument run specifications. One place to find these is on the support page, linked under the product information. You can see it in the lower right. So let's go ahead and click on that. And as I'm mentioning that, let me tell you one other way to find the specifications information. This is some of the most important information for a lot of our instrument users. Another way to find it is actually from the product page for the instruments. It's also, there's going to be a specific specifications menu within the submenu. Okay, coming back along, if you click through the specifications page, here's the kind of information you're going to see. So for the specific instrument, there's going to be different kinds of consumables, different run lengths. This will show the different data output in gigabases or reads, the runtime for different run lengths, and other really good information that's going to be helping you choose what kind of flow cell to use or what run setting to use across your different projects. And we have this for all of our different instruments. So we covered a lot of the important information for our instrument side. Let's get back to our library preparation support pages and see what kind of information lives there. Let's go back to support.illumina.com, again to our support center. And this time, we're going to look at the product category and pick library prep kits. Once you bring that up, you're going to have different kit families where you can select a specific kit from. My favorite kit of the day is Illumina DNA PCR free. So I'm going to go ahead and go to the Illumina dropdown, and then I'll find that DNA PCR free kit in the list and click through to get back to that support page. The instrument and library preparation support pages do have similar information, but it's a little bit different as the workflows are different. Let me focus on some of the very important resources for library preparation that you're going to want to review before you use the kit for the first time. Again, we're first brought to the overview page, and you can see the different options up there along the submenu tabs. With library preparation, often the most important thing is making sure you know where the protocol is. So if we go ahead and click on the documentation tab, we're going to find the kit reference guide. That's what we call our protocols. This is going to have all the information for using the kit, including the detailed instructions. I recommend that you download the reference guide and review it before your first use, preferably even before you start pulling reagents out of the refrigerator. Sometimes you have to order additional consumables. The consumables and equipment that you're going to need to provide as a user are going to be detailed in the document just below the Illumina DNA PCR free consumables and equipment list. Some of our kits come with all of the consumables needed in the catalog number you ordered, and some require a few extra parts. So make sure to look at that at the time that you're ordering the kit. This might include things like magnetic beads or a specific enzyme. For equipment, usually we'll have a validated magnetic bead stand and different thermocycler models and so on. Okay, let me do one more pulse check for our library preparation users. Thumbs up if you are able to find this page with the reference guide. Again, for library prep, this is the single most important resource for using any of our library preparation kits. Okay, we've got one thumbs up so far. Let's see if we can get one or two more. Oh, and we got a bunch more. Great. I'm glad you're all following along. Okay, what other important information lives on the support page? For both instrument consumables and library preparation, we're going to have a contents and storage page. This is going to show great info, like how many boxes come with a particular kit catalog number, the different types of tubes and the number of tubes that are in the different boxes, and where you need to store these. One thing to keep in mind is sometimes the shipping condition is going to be different than the storage condition. So you may receive a box at one condition, but need to put it in the freezer or refrigerator upon receipt. As I mentioned, some catalog numbers also are going to come with multiple boxes, and the different boxes may be shipped and stored at different temperatures. This information is also noted on the actual box and tube labels, so you always have a different place to check. Now, what if you'd like some additional training before you go ahead and plan to use the kit? We have a training tab that's going to have in-depth training videos and also curated tech support webinars for the majority of our kits. I recommend viewing one of these before the first time you perform your first prep. Our kits have different chemistries, there are different little nuances to be aware of, and it's really helpful to let you feel confident before you go and tackle that first prep in the lab. Another great way to find those training resources is to go to the training landing page. This is found at the link at the top, support.illumina.com/training.html. I particularly like this page because this is where our tech support webinars are going to live as well. You can use the filter on the left-hand side right over here to search by product, or if you want to stay abreast of the different webinars we're going to have, you can also look for recorded now on-demand webinars, and you can select the upcoming webinars if you want to specifically see the planned live webinars that are going to be given over the next few months. Like today's webinar, this is a great chance to keep aware of the different technical information that Illumina has, and also have a chance to ask live Q&As to your tech support team. Here's all the different training types that we have available through this channel. We're going to have online training. This is curated from our actual Illumina training department. It includes 10 to 20-minute self-paced courses you can take. We have support shorts, which are several-minute long, really short videos that are going to explain a really specific process that might be helpful if you're trying to do something specific to get a MySeq run going. We have these longer support webinars that go into great depth on a single topic. We're also going to have video tips from our Illumina support staff, including our application scientists in the field. This could cover things like best practices for quantifying libraries, what to do with libraries of different fragment sizes, and we even have some video troubleshooting tips for different instrument-related issues. Many of these resources can also be found directly on Illumina's YouTube channel. That gives you the general high-level view of our support website, but let's keep looking at some of the specific tools that you can use. Now I'm going to look at how you can best use your MyIllumina account. MyIllumina is going to live at another link, my.illumina.com. I recommend, if you haven't already, to go ahead and create a MyIllumina account. You're able to do this by looking at these top menu items. This will allow you, if you click on over here, you're going to have the option to register for an account. If you think you already have an account, go ahead and sign in. This will let you follow along with me for today's webinar. Now, by creating a MyIllumina account, you're going to have access to product pricing, personalized dashboards for your orders. If your instruments are linked to Illumina's system, you'll even be able to see your own instrument run history for your account. And you can see your personalized support case history with tech support, all for free. I do recommend signing up with your institution or company email address, which is going to give you better visibility and quicker access to this information. We also have different email notification options, so you can be contacted as much or as little as you prefer. Let's look at some of the specific resources you have access through only through MyIllumina. It has some what I think are really important support resources for you. So when you first sign in, you have the option to go to your account dashboard, and there's going to be these different sub-menu items. If we navigate over to the far right one, the Tools dropdown, let's first look at the Product Lot Tracker tool. You can see there's going to be some other good tools like certificates of analysis, which are for IVD products, a custom products option, MySeq self-service, and additional tools. But let's go ahead and look at Product Lot Tracker first. Okay, I'm going to send one more pulse check to see if you've found Product Lot Tracker. This required that you signed into your MyIllumina, so we'll see if anybody's able to go and find the Product Lot Tracker today. If you're not, I would suggest you go ahead and open another browser window to remind yourself to create a MyIllumina account after the webinar. Okay, we've got a couple of people. Thank you very much. Let's look at this tool, there's going to be 3 different tabs. Some of our most important information lives here. If you're a lab that needs to document your certificates of conformance for the products you're receiving, we have a tab here. Keep in mind, we don't provide certificates of analysis for all products. We provide certificates of conformance for all of our research use only products. So that's a little bit of a difference. And you're able to access these and download them if you have your packing slip or other sales information. On the right-hand side, there's a tab to pull related serial numbers if you have a particular box or tube component barcode. And then my absolute favorite resource on the website is our lot expiration tool. This allows you to pull expiration dates and related lot info if you have just a little bit of information. If you have the box lot or the component lot, you're able to pull information for that item as well as everything in the box. Now, if we go back to the dashboard, we have a specific tool that's really useful for our MySeq users. You would go to the Tools dropdown and now select MySeq Self-Service. For our MySeq users, this is going to take you to a tool to generate a MySeq RFID override code. Now, occasionally when you're setting up a run, usually this is going to be late at night, that's the way it goes, right? But occasionally a particular MySeq consumables RFID tag might not be read for some reason, and you're going to need to generate an override code to proceed. You can certainly get codes from tech support during business hours, but you can also generate them by yourself using this tool. You're only going to need to be able to enter your instrument serial number. For MySeq, that's going to start with M as in Mary. And then this is going to be giving you a code that you're going to be able to enter directly into your MySeq software to be able to proceed with the run. Now, do know if you're a MySeq DX user, you are going to have to contact tech support for this information. But our contact information is right here to make it easy for you if you forget that. While I'm on the topic of MySeqs, let me mention one additional resource that does not live in MyIllumina. This is a little bit of a tangent, but this is a new resource and I want to make sure to highlight this. We have a new MySeq support self-service guide. It's hosted at the link above, the illumina.pagetiger link. And this is an amazing link if you want to go ahead and bookmark it. It's an interactive guide. It's 80 pages, so it has to be interactive. That's going to let you easily search for information on most MySeq questions and troubleshooting topics. This is really new. You may not have seen this. I encourage you to take a look at this to give yourself better self-service options for your instrument. Let's look at another resource that lives on the website. Illumina has a customer forum called the Community at Illumina. You can access this resource from our website or by going directly to community.illumina.com. Once you're there, you're going to have the option to search for different topics. We have product type specific forums. And this, as a customer, is a great place where you can really get more information on your area of interest. You can subscribe to different product boards, and then you can interact with other Illumina users. You can get and give feedback and participate as much or as little as you like. Here's a little bit more information on that. And if you have a problem that you can't get an answer to, then also can feed into tech support as another resource for you. Our website also has a number of different interactive tools that can help you better plan your experiments and sequencing runs. You can find most of our tools back on the Support Center. If you want to go directly to this link, this is going to pull up a summary page of the most popular tools. There are some additional product selection and experiment planning tools linked at the bottom. Okay, it's been a while since I had a pulse check. I'm going to send one right now. Thumbs up if you're able to find the support tools. This page is a good one to bookmark as well. There's going to be a lot of tools that you might find useful to your particular needs. Okay, I'm waiting for a thumbs up to see if a couple of you have navigated along as well. Great, thank you. On this page, some of the highlights are we have a library prep and array kit selector, which is a more in-depth tool than that first filter I showed you earlier in the webinar. It's going to recommend kits based on your experimental conditions. We have tools for custom panel design and use if you need to pursue that option. We also have the custom protocol selector, which is a tool that can actually merge protocols across our different workflow from a particular library preparation reference guide and a sequencing system guide or denaturation guide and merge them into a single document. I do like to recommend this for our experienced users. I think it's useful to look at all of the guides separately first to understand what you're doing and then put them together. Okay, and let me focus specifically on one tool to give you an idea of what some of these can do. This is the sequencing coverage calculator. If you go ahead and click through from that summary page to the coverage calculator, you end up on a landing page that's going to give you different options. You can start with DNA, RNA, or combined applications. And then once you pick, so I've picked up the DNA application, there's going to be a drop-down that's going to list common applications or even specific products. So let's say I want to figure out how many whole genome sequencing samples I can put on a particular run. Depending on the kit, you're going to be asked to fill out different conditions. Some of our Illumina library preps will default to certain conditions. For this WGS, the default is asking for 50x coverage. We're assuming about 2% duplicates, and it's a human genome, so it's going to be having a larger genome size to cover. I looked at my instrument kit that I already had in the lab. It looks like it's a 2x150 run, so I'm going to enter in 300 total cycles that'll be covered in the sequencing reads. And then I have the option to run on a NextSeq or NovaSeq today. So I'm going to see how many samples I can put on and what flow cell type might be the best for this particular experiment. Now it's going to output all these different conditions. This seems like a lot of information at first, but let me point out what you want to focus on. First, if you want to double check the calculations, you can look at explain the estimations up here to see some of the formulas and assumptions we're making to make sure it applies to what you want to do. And then if we look down at the table, I find the most pertinent information is down here. It's samples per unit. And it can be shown either as by flow cell or by lane for some of our large or high throughput instruments. And so to run a human sample at 50x coverage, looking at the NextSeq, it's telling me I can put 0 samples per flow cell. So what that means is it's not a great idea. I need a sequencer with higher throughput to be able to get that kind of coverage. So then if I look at my NovaSeq, I can look at the different flow cell options between the SP and S4 flow cells, and I can see if I just have a couple of samples, I can use a smaller flow cell. If I want to do really high performance, high throughput, I can put up to 17 samples on 17 human genome sequencing samples on a NovaSeq S4 flow cell. So that's probably what I'm going to use for a larger project. You can also download these results to save them for later. Another really common question we get is where our safety data sheets live. You can find those by going directly to the link support.illumina.com/sds.html. And so for this one, it's going to take you to another tool. All you need is the product number that's going to be found on your box or tube label. And you can enter the product number, enter the region you're in, and we even have the option to pick different common languages for that region. You'll go ahead and hit search, and it's going to bring up and let you download all the safety data sheets that are relevant to that box or individual tube that you pulled. Okay, that was a lot of different resources I covered. I hope that's given you a great overview of the website. But you have one other critical resource that we really want you to use to enable your experimental needs, and that's our tech support team. It's really easy to reach your Illumina tech support team. If you're on the website, you can navigate to the bottom of any website page to find a link that brings up our contact page and business hours. So here I've scrolled down to the bottom of the page. I'm going to click on the Contact Us, and it's going to bring up your region's contact information. So you can see when we're available by phone or email. You can also choose to email us. It's really easy, techsupport@illumina.com. I do recommend if you have an urgent issue, let's say you're stuck in the middle of a library preparation or you're setting up a sequencing run, it's best to call us if it's during business hours. That way we can help you in real time. Now, tech support covers all of Illumina's commercialized products. We're broken in the Americas region into some specialists so that we can better cover our products. So we have a team that focuses entirely on our microarray workflow, and you can see the common types of inquiries and troubleshooting questions we might get listed here. On the sequencing side, we're broken into 3 workflows to better support you. We have our sequencing library preparation group that's going to support any questions around experimental design and using our Illumina library prep kits. We have a group that focuses on our sequencing instrument operation and run performance. So if you're setting up the run, if you have a question about your instrument, if you think there's a problem with the instrument, this group's your first place to go. And then we also have a sequencing data analysis software group. Sometimes they do get involved in experimental design as well. They know all the details about our different software and can better advise you there. Do you have a few tips about how you can more quickly resolve your questions when you're working with tech support? If you have a really specific question or an issue you're looking at, often it is useful if you're able to provide details at the time you're contacting us. As an example for library preparation, if you have some questions on your final library, it might be helpful if you're able to provide some quality information such as fragment analyzer or bioanalyzer traces and the concentration info and how you quantified that. For sequencing or analysis, that might mean being able to provide the sequencing metrics or analysis results, perhaps sharing a link to BaseSpace. This can enable us to more quickly take a look at what's going on and get a direction to go with you. Now, often for more complex inquiries, we may need more information, and it's helpful if you're able to provide that requested data or know when you might be able to. Sometimes I know you may already have an idea of what happened, but we often like to double check that there's not something else going on so that we can resolve it the first time. It's also good, some of these things get complex, so it's good if you're able to respond in a timely manner. If you know it's going to take you to get a while, it's going to take a while for you to get the data, it's helpful if you can work with your tech support team on when you want to continue working on this. Sometimes you might have to go to a colleague or another core lab to get more data, and it's helpful for everyone to have the same expectations to be able to better support you. And then finally, every time you report a new question or issue to tech support, it's going to generate a case number where we can take notes on your case so that we can help you better the next time and understand your particular account and individual history. If you're coming back to a case after a little bit, it's helpful if you have that case number handy so we can go back to that previous work. Now, if you call in, we have the really great option to share your computer screens with you. We have a site that's share.illumina.com where you're going to be directed to download a really small piece of software to enable this sharing. Do keep in mind that anytime you're using a new instrument or a new computer, you are going to have to download the software. One quick tip is if you just had an instrument installed in your lab, it's great to go ahead and install this software now before you need it. That way, if you're calling in when you're in the middle of the run, it's a little bit less stressful. We have another way of sharing a lot of run and analysis data if you're using Illumina's BaseSpace Sequence Hub platform. This is our cloud run monitoring and analysis platform that lets you share information both with your colleagues and your Illumina support. If you already have a BaseSpace account, I'm going to give you a little bit more homework. Go ahead and try to sign in now so that you can follow along as we generate a link to share. If you don't have a BaseSpace account, you might want to wait until you're ready to do your first sequencing experiment. You can get free analysis credits during a trial period, so this is a great resource for you to use in the future. Now, I've already signed into BaseSpace on my screen here, and let's say I want to share a sequencing run with tech support. I'm going to go to the dashboard, and I'm going to first click on Runs tab. I want to share a run today. Once I've looked at my runs and selected the one I'm interested in, so here's the NovaSeq S4 run, I'm going to look at the submenu to see my share options. And so if I look at this option, the 3 dots connected by lines, it'll give me different options for sharing. I can even transfer the run to another colleague. When you're working with tech support, we want you to go ahead and use the Get Link option at the top. This is going to generate a shareable link that will allow anyone that you email the link to go ahead and access your run. We prefer that you don't send an invitation directly to tech support at illumina.com as it's not monitored, and we wouldn't be able to share it across our colleagues if we need to consult with another specialty. So again, it's best to use the Get Link feature to get a copyable link that you can put in your email to us. And the great thing about this is you can disable that link. So whenever we're done working together, you can disable the link and be confident in your data privacy. You have the same functionality for your analysis projects. So if you click through the Projects tab, you can also utilize the Get Link option to share with tech support. Finally, I'm going to advocate for an even better option. We now have Illumina Proactive. This is an option to connect your instruments to our cloud monitoring, and this is going to get you the absolutely most efficient support. If your sequencing instrument is hooked up to Proactive, Illumina is able to see the high-level run metrics and log files, so you don't have to waste time providing these files anytime you're working with us. On some instrument types, our software can even start to proactively diagnose future instrument issues, which can alert you for the need to fix before it impacts your workflow. As a benefit to you, you'd also be able to see your run metrics and high-level history in your own MyIllumina account dashboard. If you're interested in learning more about this specific offering, we have a lot of information on this on the website, which covers the benefits of this free service as well as our data security policies. And you can also find it if you just want to search for Illumina Proactive within the website search bar, it'll pop right up. Okay, thank you so much for taking the time to join me today. I hope this session has been helpful